Hello, and thank you for joining us for this presentation today. I will be presenting an introduction to the analytics of cell and gene therapies with a primary focus on AAV. In addition to my own data, I will also be presenting data from my colleagues Jimo and Hua. Here is a brief outline of what I will be discussing today. I'll start with an introduction, a very brief introduction of gene therapy and analytics. Then I will cover the analysis of AAV capsids by several LC and LCMS methods, and I'll go into those in a little bit more detail later, and then I'll finish up with a summary. We can see, based on the chart on the left, that there's been tremendous growth and interest in using AAV vectors as part of gene therapy. Along with that increased growth, we see increased information provided by regulatory agencies such as the FDA and others, as shown on the right, which basically help us to determine what we need to understand about the structure and function of these compounds in order to ensure that they are safe and efficacious for patients. Shown on this slide is a tabulation of the LC and LCMS technologies that we will be discussing today for the analysis of AAV. In terms of capsid purity relative to aggregation and fragmentation or high molecular weight species and low molecular weight species, this is traditionally done by AUC. While a very good technique, AUC is a low throughput technique with long sample turnarounds. We will be looking at the use of SEC with FLR or fluorescence detection for this type of analyses. In terms of capsid content, traditionally ELISA is used. While this is a very high throughput technique, it does suffer from sample to sample variability as well as concerns about controlling the reagents of the actual method. By contrast, we will be looking at the use of SEC with both fluorescence and UV detection in line to monitor the amount of capsid that is in a particular sample. In terms of the purity of the sample, one area of consideration is the empty-full ratio, or the amount of capsid that contains the requisite amount of single-stranded DNA, which is important for the function of this particular therapy. This is traditionally done by AUC, and we will look at the use of anion exchange chromatography, which will measure the difference in the surface charge profile and separate the empty and full capsids based on that. Again, in terms of purity, relative to the presence of other proteins or variations in the VP1, VP2, and VP3 proteins that make up the capsid structure, traditionally this is done by SDS-Page. We will present data using reverse phase LC with MS detection to monitor these particular changes. And finally, in terms of capsid identity, this is traditionally done by Western blots. It's resource intensive and takes considerable skill in order to get good results. We will look at this type of separation or this type of analyses using peptide mapping for capsid identity, while also obtaining significant information about amino acid sequence and post-translational modifications. Now we will look at the analysis of AAV capsids by size exclusion chromatography. First, we will discuss some of the principles of size exclusion chromatography and how they relate to the separation of large particles such as AAV. It's important to remember that the separation is based on the size of these particles in solution, or their Stokes radius. Ideally, there's no absorption of these particles to the surface, so we have to go through some method optimization. And ultimately, the pore size and the pore volume of our column are going to really be the critical parameters that drive this separation. We also want to remember this is a very low resolution technique compared to other methods such as reverse phase, where we have very large peak capacities. Typically for SEC, we'll be dealing with peak capacities in the order of 10 or so, and as these analytes get larger, those peak capacities will actually become even lower. To understand how this separation occurs, let's look at the middle figure. Here we have two analytes, a red and a blue analyte, with the red analyte being significantly larger, or the dimer, and the blue being the monomer. We can see that the dimer is excluded from some of the small pores, but even when it gets into some of the larger pores, it can run into these constrictions in the pore structure, so it can't proceed and diffuse into any of these other pore volume as shown down here. So ultimately, because there's less pore volume for this larger molecule to diffuse in and out of, it will spend less time in the pore structure of the particle, more time in the flow stream, and it will come out sooner. Whereas the smaller particle can go into some of the smaller pores and it can pass through some of these constrictions. And it will basically spend more time diffusing around inside the particle and less time in the flow stream, so it will come out later. As I mentioned previously, SEC separations are driven really by the size of the analyte as opposed to its molecular weight. For example, if we compare the molecular weight of a monoclonal antibody at 150 kilodaltons to that of an AAV at 4.8 megadaltons, the AAV with its single-strand DNA is about 32 times greater in mass than that of the monoclonal antibody. However, when we look at the capsid structure of the AAV, it is only about 50% greater than the hydrodynamic radius of the monoclonal antibody. Therefore, we feel that generally we can drive these separations based on our SEC separation technologies currently available. However, as we get into other types of gene therapy vectors, such as lentivirus, where the sizes are now getting into the 100 nanometer size, those are no longer going to be capable of being separated by the technologies that we're talking about here today. I wanted to point out a couple of the tips and tricks that I found as I was developing SEC methods for these AAVs. In terms of method optimization, remember we want to minimize secondary interactions. I found that using potassium chloride was more effective than sodium chloride for minimizing secondary interactions, and I didn't find any significant benefit in using some of the other additives for these type of separations, such as arginines or organics. I also want to point out that the use of fluorescence detection over UV is certainly recommended. By using intrinsic protein fluorescence with excitation at 280 nanometers and emission at 350 nanometers, we can extend our sensitivity. However, you want to be aware that the actual measured high molecular weight levels will be different for UV versus fluorescence, since the principles of the absorbance versus the principles of fluorescence can be really changed by environmental conditions surrounding those particular chromophores or fluorophores. We wanted to get an understanding about what the size limits of our BEH 450 angstrom SEC column might be. So we had some previous data showing the separation of IgM, which is a very large complex composed of 5 antibodies in a ring. And generally, it's been shown by electron microscopy data to have approximately a diameter of 40 nanometers or so. And we can see that we can separate out that pentameric version from the dimer of that, or the dipentameric version. This therefore gave us confidence that our 450 angstrom SEC column may provide a useful separation for our AAVs. Shown on this slide are the separations of 6 different AAV serotypes, 1, 2, 5, 6, 8, and 9, on the BEH 450 angstrom SEC column. The variations in these separations really relate to the amount of potassium chloride that was necessary in order to get good peak shape and also see our peaks return back to baseline. We also see in these separations that we can separate out multimeric as well as dimeric forms of aggregates for some of these particular serotypes, as well as fragments for some of these particular serotypes. Next, we want to discuss the analysis of capsid content, or CP per mil, using SEC as an online buffer exchange with fluorescence detection. Remember, this is typically performed by ELISA. Ultimately, there are significant optical challenges that we have to consider when we do these type of analyses, and the size of the particles is approaching the wavelength of light that we're using, such as light scattering. So getting our analyte in reproducible mobile phases or reproducible buffers is incredibly important. I also had this idea based on looking at a method that was published recently for anion exchange separations of AAVs, in which the authors showed, as shown below here, that the response factor, or the amount of signal that you get for an empty capsid versus a full capsid, is much closer to 1 for fluorescence than it is for UV absorbances. Therefore, what we did was looked at a dilution series of AAV capsids that were about 50% full, and we can see that we get linear responses for the fluorescence as shown in orange, as well as for the UV absorbances as shown in gray and blue. Now, these empty-full ratios still have to be corrected for these samples. We can imagine that if they shift wildly, we're going to get significantly different responses for the UV and maybe not so much for the fluorescence. These can be determined by either anion exchange chromatography or analytical ultracentrifugation. However, since the response factor is very close to 1 for fluorescence, we wanted to investigate whether we could just approximate empty-full ratio using the A260, A280 peak area ratios, and then apply that for our fluorescence quantification by SEC. As mentioned on the previous slide, we have to make a response factor correction for our fluorescence. And I wanted to briefly talk about how we're going to estimate the empty-full ratio based on the A260, A280 peak areas in order to apply that response factor correction. Shown on the left are chromatograms using our BEH 125 SEC column, and this is a short column, 4.6 by 30 millimeters only, so we have about a 2-minute run time. And we can see that we get good signal to noise for FLR, and a little bit less signal to noise in terms of the A280 and A260, but adequate in terms of determining these relative peak areas. If we take the A260 and A280 peak areas, as shown here, and we make a ratio out of those and plot those versus the mole fraction of the full capsid, so this is relative to the empty full ratio, we can see that we get this fit to a curve that follows this equation. So then ultimately, for our unknown sample, if we can measure the A260, A280 peak area, then we can interpolate the mole fraction for that and the empty full ratio and apply that correction for our fluorescence response factor. Next, I will talk about the separation of empty and full AAV capsids by anion exchange chromatography, and this is work that was performed by my colleague Hua. There are a few analytical methods that are commonly used to analyze the full empty ratio of AAV capsids. As previously mentioned, AUC is really the gold standard for this measurement, and it has the advantage of being able to measure partially filled, or capsids that have only a fragment of the single-stranded DNA within them. Another commonly used method is spectrophotometry, which involves disrupting the capsid and measuring the UV absorbances at 260 and 280 in order to infer what the empty full ratio is. While this method is less time consuming and fairly high throughput, it can suffer from optical interferences. As we'll discuss here today, ion exchange chromatography, in particular anion exchange chromatography, can also be used. This uses significantly less sample, but does require method development. The separation, as shown in the middle of the slide, is really driven by the presence or absence of single-stranded DNA within the capsid, really changing the overall surface charge of the capsid. And that separation, as shown further on the right with the chromatograms, can really cause the separation of empty and full capsids from one another. Another option, as more of an investigational technique currently, is charge detection mass spectrometry. This also uses less sample and doesn't require significant method development and has the capability of measuring partially filled capsids. But clearly this is much more of an experimental technique at this point. Shown on this slide are the results for the optimized AX separation for AAV8. We can see that we have a nice response and a nice separation between the empty capsids and the full capsids using fluorescence detection. We can also see, as shown on the plot on the right, that we get a nice linear response for the measured percent empty versus the predicted percent empty for this sample using the equation shown in the plot to correct for the response factor of the empty and the full. I do want to point out in this separation that if we look at the green trace, which is essentially 100% empty sample, we see some charge heterogeneity that is co-eluting with our full capsid. The A260, A280 UV absorbance ratio doesn't shift for this empty trailing part of this peak for the empty capsid. Therefore, we presume that this is charge heterogeneity of the surface proteins of the empty capsid. What these data demonstrate is that while we would hope that we could separate out partially filled capsids in between the empty and the full, because of the charge heterogeneity of the empty, we don't believe that will be possible for this method. Shown on this slide are the separation of a series of different AAV serotypes using the optimized conditions developed for AAV8. We can see that the retention times can vary quite significantly. And what we can understand from this is that in order to develop an anion exchange method for empty-full characterization for the other serotypes, method development will have to occur using a sample that is essentially an empty sample and a sample that is essentially a full capsid sample in order to work through that method development. I will next talk about the characterization of the capsid proteins by reverse phase liquid chromatography and LCMS as performed by my colleague Zemo. Next, we'll discuss the analysis of the three proteins that make up the AAV capsid protein shell. This protein shell provides protection to the therapeutic genes from the environment and also has a direct impact on viral infectivity. As shown in this figure, the viral shell of AAV is made up of 60 proteins, 50 of these being VP3, with the sequence as shown in blue, and if we extend that sequence, we get VP2, which AAV is comprised of 5 of those, and if we extend VP2 further, as shown in gold, we'll get VP1, and there's 5 of those. The challenges of analyzing AAV are that the concentration is generally much lower than typical antibody or other protein separate solutions, to the point that some of these preparations of AAV are 10 to 50 times lower in concentration than that of, say, a typical antibody. In addition, these formulations typically contain different surfactants that can cause trouble for our analyses. But the advantages of developing an LCMS method for the analysis of VP proteins in our AAV is it gives us more data, as shown on the right, versus running a gel in terms of looking for impurities or getting additional structural information from those VP proteins. In this slide, we demonstrate the importance of including some sample prep when we are analyzing the capsid proteins of AAV by LCMS. In this particular sample shown in the top chromatogram, we have an AAV sample that is formulated with a surfactant. As we can see, if we do not remove the surfactant, we can see a rise in the baseline on our total ion count from our mass spectrometer. If we perform a sample pretreatment that removes the surfactant, we can see that we make that baseline rise less significant. And as you'll see on the next slide, that will have a significant impact on the data quality that we get from the mass spectrometer. Shown on the left of this slide are the LCMS chromatograms for the samples both before and after we remove surfactant. And shown on the right are the mass spectra obtained from those LCMS runs. We can see that as we remove the surfactant, as shown by the bottom spectra, that we reduce the noise significantly and increase our signal quality. In addition to sample prep, we also need to develop a good LCMS separation and have the right tools to enable us to know if we're getting the separation that we need. This work was performed by Zemo on a bioaccord system using an RDA TOF mass detector. Shown on the right is some of the separation development. Here we see that by using difluoroacetic acid versus formic acid for the separation, we get better peak capacities and better separation of our VP proteins. In addition to mobile phase selection, the selection of the column that we use for the separation can also be a critical consideration. And this middle chromatogram is the chromatogram we saw from the previous slide. However, there is a pair of proteins in the peak at 13.2 minutes that we desired to have separated from each other. We can see that we can't quite separate those on a C18. However, if we go to a shorter alkyl chain length, then we can get that separation to occur on our C4 column. However, for a different serotype, AAV5 instead of AAV8, as we were observing before, we can see that a C4 column may not be the optimal choice. And in this case, we can see that there are some unresolved peaks that are occurring on our C4 with DFA as our mobile phase. However, now with this particular serotype, if we go to C18, we can pull out the peaks that we are interested in. Once we have the separation determined for our VP proteins, we can go ahead now and get high resolution mass spec data for those proteins. And in this case, these data are collected using our bioaccord TOF system. And we can see that for the VP1, VP2, and the variations of VP3 proteins that we can obtain mass spectra that allow us to make those identifications. We can also obtain some important post-translational modification data from these LCMS runs. As shown here for VP1 and VP2 proteins, we can see evidence in both of these mass spectra that there is phosphorylation of this protein. In addition to obtaining MS data for the VP proteins, we can also use the UV trace of the LCMS run to monitor the abundance of the VP1, VP2, and VP3 proteins that are in our sample as shown in this slide. Similarly to what we observed by size exclusion chromatography as well as anion exchange chromatography, using intrinsic protein fluorescence can also be a benefit when doing these LCMS analyses by increasing the sensitivity and decreasing the background noise and baseline drift of the separation as shown in this slide. Shown on this slide are the separations of 6 different AAV serotypes as separated by these LCMS methods, showing the identities of the peaks identified by the bioaccord system. Next, we'll talk about LCMS peptide mapping work that has been done by Zemo for the analysis of the AAV capsid proteins. In speaking with many of our customers, while the necessity of having a peptide map as part of product release is debatable, it is clear that having a peptide map available to help with product development and probe small changes in AAV structure is going to be important. Ultimately, an LCMS peptide map can confirm our amino acid sequence for our capsid proteins, as well as help us discern degradants such as oxidation, deamidation, and also post-translational modifications such as phosphorylation and glycosylation if those are present. It can also be very helpful in understanding what the identities are of some of the host cell proteins that may be in our sample preparations. The general procedure for peptide mapping will be to take our AAV capsids, dissociate those into their respective VP1, VP2, and VP3 proteins, and then digest those and run our LCMS. When developing a peptide map for the VP proteins of an AAV, there are a number of challenges and considerations. First are challenges in the sample. We have very limited sample concentrations and amounts in terms of micrograms per mil or micrograms. We also have surfactants generally in our formulations, which can really make it difficult to get good LCMS data. In addition, the samples itself, in terms of the VP proteins, as I've discussed, there's significant overlap in the amino acid sequences as shown on the right. In terms of addressing these, there are two options. One is digest the pool of proteins, the VP1, VP2, and VP3 together. Ultimately, this will lead to uncertainty as to which proteins are modified, and it will also require a very wide dynamic range to detect the VP1 and VP2 variants. This ultimately will require LCMS detection in order to identify and even track those particular variants due to the large dynamic range that's going to be needed. We could also isolate the VP1, VP2, and VP3 proteins and peptide map those individually. This would add significant complexity and time and result in even greater sample consumption due to lower recoveries. As a result, we've looked at this and we've determined that we are going to develop the peptide map for the pooled proteins. Ultimately, our goal is to develop a reproducible digest for micrograms of this protein and provide a reproducible and reliable LCMS analysis. Ultimately, the most effective peptide mapping procedure developed so far using only 2.5 micrograms of protein or capsid as starting material has been to disrupt the VP proteins of the capsid and then send those proteins over an SEC column for sample cleanup. A collective fraction representing the VP proteins is then collected and then subjected to trypsin digestion and mapped as shown here. In evaluating the LCMS data from this sample preparation, we have observed greater than 90% sequence coverage for the VP1, VP2, and VP3 proteins when taking into account miscleavages. We are currently working on optimizing the digest conditions to reduce those number of miscleavages. In addition to the sequence coverage, we've detected a number of modifications, including the site-specific phosphorylation, as well as deamidation, oxidation, and N-terminal acetylation. In summary, we have seen that the BEH 450 angstrom pore size SEC particles are well suited for the aggregation and fragment analysis of AAV. However, due to the pore size and particle size of these particles, these will not likely be able to address the analysis of larger viral vectors such as lentivirus. We have also seen that SEC UV FLR in tandem can provide high throughput measurements of capsid concentration. In terms of anion exchange chromatography, a method was optimized for AAV8 empty full analysis, but we have seen that it will not likely provide for the analysis of partially filled forms of the AAV. The method will also likely need to be modified for other serotypes. In terms of the reverse phase LCMS separation of VP proteins, we've seen that DFA acts as a very good modifier, giving good chromatographic and MS performance, along with the BioAccord LCMS system, which is very well suited for the routine LCMS analysis of AAV capsid proteins. In addition, it's well suited for peptide level analysis, as demonstrated by the final set of experiments, where we've seen a promising peptide mapping procedure using analytical SEC desalting and LCMS peptide mapping with that BioAccord system. I'd also like to let you know that much of the data that you've seen presented here today is also presented in these app notes, as well as additional information on some of the procedures that were used to collect those data. In addition, the SEC FLR UV capsid concentration application that I discussed will be coming soon, in addition to a peptide mapping application note, which is scheduled for later in the year. I would like to thank you for your attention and also acknowledge the contributions of many of my colleagues here at Waters, as well as help from our collaborators at Sanofi and BioReliance for providing both samples as well as information.